Hello, my name is Jennifer Talbert, and for the project, I will be discovering the evolution of the Apicoplast in Plasmodium falciparum pertaining to malaria and touching on some of its drug resistance factors. To begin, I just want to talk about what Plasmodium falciparum is and the importance of it. So to begin, Plasmodium falciparum is under the phylum Apicoplast in the genus of Plasmodium. Plasmodium falciparum is specifically the deadliest parasite causing malaria. However, other Plasmodium species have also been known to cause malaria at just a lower extent, and there's growing research to show that the possibility of other parasite species in general can also play a part in malaria. As many are aware, malaria is a burden on the human population. Malaria is a life-threatening disease that compromises the immune system in humans, mostly in children 2 to 10 years old, but along with in pregnant females. In the past, there has been great eradication efforts, both from therapeutic standpoints and preventative standpoints, and even development of various vaccines, such as the RTS vaccine. And according to the World Health Organization, in 2019, there was an estimated of 229 million cases of malaria, and an estimated of 274,000 deaths worldwide. Most cases are shown in African regions, which carry roughly 94% of malaria cases worldwide, as shown in the map here. Malaria's vector is the Anopheles mosquitoes, which tend to live in fresh or saltwater marshes, grassy ditches, streams, rivers, or temporary rain pools, essentially just warm, humid environments. And this is why, in general, mosquito populations tend to be more dense during warm months. Unfortunately, it takes great wealth and resources to prevent these mosquitoes, and malaria, as many people do not know, is pretty prevalent in America still, but due to the draining system that has been established in agricultural areas, along with the use of insecticides, there has been a significant decrease in malarial cases here. However, in South Africa, in the Sahara areas, and even in South America, a lack of funding and resources have put these areas at a disadvantage, and thus the mosquito population tends to be denser, causing more malarial cases. One major use to prevent malaria was DDT, which was the first modern synthetic insecticide in 1940s to combat malaria and other insect-human diseases. This provided beneficial for decreasing mosquito populations. However, there was one major downfall, and this was the toxicity to the environment and the humans, which later caused it to become banned in the US in the 1950s. Since then, vector controls are limited to individual levels using personal insecticides and home protection in terms of prevention, which we do in the summers in the US. And there is still a lot of growing research in terms of pharmaceutical treatments, which I will cover here in this presentation. So prior to the discovery of its protozoan agent by Leverian in 1880, malaria was thought to be caused by fumes emitting from swamps. However, British and Italian scientists revealed Plasmodium was transmitted through the bite of a mosquito. It is now known Plasmodium falciparum, along with the other Plasmodiums, are transmitted through the bite of an infected female Anopheles mosquito, also known as a malaria vector. Upon injection during the so-called spitting phase through the mosquito's proboscis, the saliva containing sporozoites will enter the human blood cells, or erythrocytes, and make their way into the liver and invade hepatocytes, and this is known as the infective stage. Then will undergo multiple rounds of replication, making merozoites, meaning individuals, and these affect mature erythrocytes. This cyclic cycle will continue on with replication and then apoptosis of the erythrocyte, and upon apoptosis they are known as trophozoites, which just essentially means that they're feeding on the human erythrocytes, the host. And in this case, the human body will begin showing malarial symptoms such as extreme chills and high fevers. These symptoms eventually compromise the immune system and lead to extreme illness and even death in some cases. However, at one point in this cycle, one of these sexual forms of the parasite is thrown off, making the gametocytes, which will circulate into the bloodstream until they are ingested by a mosquito during feeding. When the mosquito ingests the gametocyte, sexual reproduction will occur in the stomach of the mosquito, creating more sporozoites, and then the cycle will continue on. Depending upon the standpoint, so say in ecology, the human is defined as the intermediate host for Plasmodium falciparum, since the actual sexual reproduction occurs in the Anopheles mosquito. However, often in medical standpoints, the human acts as a definitive host due to causing pathology, and this sometimes causes confusion when looking at research articles. Plasmodium is morphologically different from most parasites. Under an electron micrograph, Plasmodium bodies look very similar to other parasites, showing a nucleus, mitochondria, plasma membrane, so forth. However, there is a large spot, which in the early 60s was just labeled as spherical body, and during this time it was hypothesized that this body was a storage area for the mitochondria. Later, though, it was discovered that this in fact was an organelle called the Apicoplast. It is a distinct four-membrane-bound organelle, likely coming from a secondary endosymbiotic event. This could have occurred in early history from a gram-negative cyanobacteria that was engulfed by a eukaryote, which evolved into red algae. Then another eukaryote host engulfed that, which thus resulted in the four membranes. Research has shown that the first membrane, the outermost, is analogous to the phagosome membrane of the host cell, and the second outer membrane originates from the plasma membrane of the engulfed red algae cell. The inner membranes are equivalent to the outer and inner envelopes of the chloroplast, and then further research is needed to know the origin of the secondary endosymbiont. In early research, it was debated upon if green algae or red algae was the original endosymbiont. However, red algae has been proven by the discovery of photosynthetic 8B complexion, chromia, which has a plastid derived from red algae and has the same origins as the achioplast. This broad hypothesis also supports the Chromio-Malveato theory, proposing all algae belonging to the Chromio-Malveato possess secondary red plastids acquired by a single common endosymbiotic event, where red algae are engulfed by a heterotrophic eukaryotic host. In the mid to late 90s, this was also proved by DNA research. Williamson sequenced the genome of the achioplast, and Jeffrey McFadden localized the genome to the organelle. The results showed a 35 kb genome, its own RNA polymerase, which was a prokaryotic polymerase. It had a super operon of translational machinery, tRNAs, and some of its own chaperones. proving its prokaryotic characteristics. This discovery had hopeful wishes for pharmaceuticals, thinking they could target this organelle specifically and not just the host. However, antibiotics have always shown resistance features, even prior to this research. In Rama et al.'s research using clindamycin treatments, which is an antibiotic affecting replication, translation, and transcription within prokaryotic cells, it showed that the antibiotic was only effective after a 96-hour period, which has been hypothesized as a delayed death phenomenon, which is why these antibiotics tend to be ineffective for acute treatments of malaria and instead are used for prophylactics. Scientists wondered how this delayed death phenomenon occurred, and Joseph Desari and Phil Rosenwald researched why this delayed death occurs. It was discovered during a transcriptic profile of malaria that there were no changes in the entire nuclear transcriptome, which is kind of different from other organisms. However, it was shown that there were transcriptional changes in the Apicoplast, meaning the apical genome was being failed to replicate and transmitted to the daughter progeny when using anti-malarial drugs. Due to this, the mutation, the new merozoites did not contain any Apicoplast, so when they entered the new cell, they would die. This research has led to the development of research pertaining to metabolic pathways and questioning overall what the Apicoplast function is in the Plasmodium. With the observed loss of the Apicoplast in parasites treated with antibiotics and the elimination of parasites with drugs targeting the Apicoplast metabolic pathway, there has been shown one or more anabolic products of the plastid being essential for the parasites to establish new infections. It is believed that the metabolic pathway of the Apicoplast contribute to lipid production and the modification of lipid-bound proteins. From this, it is gathered that the organelle most likely plays a crucial role in the successful establishment of the parasite-to-host interactions. However, even with all of this research, it is still unknown what metabolic products the Apicoplast actually makes and what their role in the parasite truly is. This is what makes it troublesome to biochemically manipulate this organelle for malarial treatments. As of now, the most recent advances have been from a new vaccine from Oxford's Jenner Institute, which has shown to be 77% effective. However, this has only been a small-scale trial, only trialing about 450 children. But there is a great need for a new vaccine since the current vaccine on the market has a low level of efficiency amongst young children and infants. As of now, in terms of prevention, the CDC, along with the World Health Organization, has provided many organizations across the globe and fundraising to supply these insecticide-treated nets in countries where malaria rates tend to be high. These nets allow for people to sleep safely during times when mosquitoes tend to feed. They greatly reduce the number of mosquitoes entering the household. However, if the household tends to be large, there is still a higher chance of malaria than not. There are also various treatments depending on the disease severity, species of parasite causing the infection, and which area of the world the individual was effective. This helps determine the probability that the organism is resistant to a certain anti-malarial drug. However, it has also been shown that there is growing resistance in many of the anti-malarial drugs, so this field tends to be growing quickly. As stated earlier, there is a lot of ongoing research regarding malarial treatments, including preventative treatments and prevention, vaccines. As we gain further insight of pathways unique to the parasites, the discovery of drug targets will continue to grow. This will continue to be a big challenger to many researchers across the globe. However, with great insight on the evolution and origin of Plasmodium and its Apicoplast, it has made this challenge just slightly easier by targeting the Apicoplast for intervention against harsh symptoms of malaria. These are the sources that I have used for this paper or this presentation. This is the current source I used for the new Oxford Jenner Institute vaccine. However, it is not quite published yet, so there's no published paper, just their research methods.